Hi everyone, I'm Coralie Muller and I will present my poster modeling the dynamics of Salmonella infection in the gut at the bacterial and the host level. The dynamics infection in the gut is a result of a complex interaction between the pathogen, the host, and the microbiome. To improve the understanding of the mechanistic processes of the infection, mathematical models can be used. Several key points have to be considered regarding the generation of such model, however. First, the model has to take into account the inherent dynamics of the infection process. A high computational cost can be associated to dynamic simulation, depending on the size of the microbial community or need for parameter inference. Specialization is particularly important in the gut, as the variability in bacterial community composition depends on its location in the gut, which impacts the microbial interaction. Specialization increases the computational cost of modeling. We are interested here in methodologically addressing these limits by providing an efficient approach for metabolic modeling in time and space. As an application, we focus on the infection of Salmonella enterica serovar T. fimarium. We construct a model of the host microbiota pathogen system and use statistical learning to speed up the computation and afterwards integrate the specialization. The host microbiota pathogen system uses three metabolic models: Faecalibacterium protenizi, referred as SPRO, as a representative butyrate-producing commensal bacterium, the enteric pathogen Salmonella, and the HISA, human genome scales metabolic model, to represent the metabolism of colonic epithelial cells. The genome scale metabolic networks, or GSMs, are the core of the numerical models used for simulation, such as flux balance analysis, FBA, that simulates the metabolism of the species under an hypothesis of quasi-steady state and a set of thermodynamical constraints. FBA computes activity rates or floods of metabolic reaction in the GSM models. Dynamics FBA integrates the flux calculated in FBA into a system of ordinary differential equations, enabling metabolic simulation over time. Several refining steps have to be carried out to build the model, but I will focus here on the two most important. Firstly, the correlation of the butyrate metabolism in the host model. This is an important function for the energy metabolism in the epithelial cells. We have created the reaction linked to the Krebs cycle, TCA, and the electron transport chain, ETC. Secondly, defining the objective function of the simulation. For the metabolic networks of Salmonella and FPRO, we keep the biomass maximization as an objective function. In the case of the human enterocyte, we observed that the butyrate concentration has no impact on the biomass production, but has an effect on the energy metabolism through ATP production. Therefore, we set up as an objective function in the human cells the production of energy through the ATP synthase of the mitochondria. In addition to the three genome-wide metabolic models, we have established a set of four state variables: neutrophils, nitric oxide, oxygen, and butyrate, phenomenologically representing other important factors in the infection. For example, the inflammatory response is represented by the neutrophil state variable, and the O2 state variable is fixed with a constant flux to represent the supply via the bloodstream. We saw here that we performed a 90-hour simulation of bacterial and epithelial metabolism, with a pathogen infection occurring after 40 hours and associated with an inflammatory process. As expected in absence of infection, butyrate production is ensured by commensal cells, FPRO, in the lumen. The growth of FPRO is ensured and its niche is stable. When Salmonella infects the lumen, the human system is triggered by virulence factor secreted by Salmonella, causing inflammation and leading to the infiltration of neutrophils in the lumen and the decline of FPRO population. This implies a reduction of butyrate production and availability for enterocytes. In this situation, the TCA cycle and the ETC activity via butyrate is blocked, and the energy metabolism of enterocytes switches to the secondary pathway. The ATP production is noticeably reduced, and so is the consumption of oxygen in the cells, leading to an overflow of this molecule into the lumen, which was beforehand hypoxic. This creates a favorable oxidative niche for Salmonella growth by the oxidation of metabolites. At this stage, we have a dynamic model of the host microbiota pathogen system that accurately represents the molecular processes involved in the infection of Salmonella. About our meta models, we call that simulation of DFBA models implies the computation of metabolic flux at each time steps of the numerical integration chain. We would like to bypass this computation by approximating the mapping that takes as input the flux constraints and returns the optimal flux given by FBA. The approximation, also called metamodel or surrogate model, can be obtained by the use of machine learning techniques. We also plug the recently correlated surrogate models in the numerical integration chain and compare the computing time and precision with respect to the DFBA simulation. At the moment, we are able to derive surrogate models that speed up the system by a factor of 45 with a relative error of less than 5%. In conclusion, we succeed at efficiently capturing and predicting the metabolic roles of each partner in Salmonella infection, able and efficiently compute surrogate approximation of the model at the cost of limited loss in accuracy. An originality of our work is the integration of the human metabolic model in the system. In a previous work, we used a phenomenological description of enterocyte butyrate metabolism. Here, the model is mechanistic. Our model paves the way to a more systematic use of metabolic models as descriptor of microbe-microbe and host-microbe interaction during infection. The perspective and current developments of our work focus on optimizing the speed-up and the accuracy of the surrogate computation. We are also in the process of building a spatial model of Salmonella infection. And a broader perspective would be to generalize such approaches to other ecosystems and other infections in which metabolism play a crucial role for the dynamics of the community. Thank you.